Hi everybody, today we're going to be talking about mitosis and meiosis. If you have not yet watched the cell cycle video, please go back and watch that first and then come back here. So today we have to talk a little bit about chromosomes before we get into the phases of mitosis and meiosis. In this nucleus that I have drawn, I have placed two chromosomes. And you can see that one chromosome has 3 beads on either side of the magnet, and this chromosome has 2 beads on either side of the magnet. In our model, we're using these beads to represent genes and the magnet to represent the centromere. Now, in a real chromosome, there's not just 3 genes on a real chromosome. There would be thousands of genes on a chromosome. But chromosomes are named for how long they are. So chromosome 1 would be longer than chromosome 2, and that's how we've set it up here. We have chromosome 1 is slightly longer and chromosome 2 is a little bit shorter. In humans, the number of chromosomes is 23. So we would have to have 23 of these to represent a human cell. So we're not going to do that. So we're just going to use 2 chromosomes right here. And we're just going to say that our organism that we're playing with has an N of 2, N being the number of chromosomes. In a diploid cell, and these are your general somatic body cells, the number of chromosomes is 2N, which means you have 2 copies of the number of chromosomes. 2 copies of the number of chromosomes equals 4 chromosomes. So we have to put some more chromosomes in here. So we've got 1 copy of chromosome 1 that came from your maternal parent, and here's another copy of chromosome 1. We have 1 copy of chromosome 2, and here's our second copy of chromosome 2. So this one came from the maternal parent at fertilization, and this one came from the paternal parent at fertilization. Same thing here. And you can see that these guys are similar. They have the same number of beads, top and bottom, but they are slightly different, which is why we put them in a different color. So while this bead might represent eye color and might be coding for brown eyes, this bead here would also represent eye color, but might be a slightly different version of the gene that codes for blue eyes. So these two are similar, but not genetically identical. We call those chromosomes homologous chromosomes. So in all of your general body cells, any cells that are not sex cells, you have this setup. It is diploid, and you're going to have 2 copies of every chromosome. So remember when I said in humans, N equals 23? Well, you got 1 set of chromosomes, that's 23 from your mom, and 1 set of chromosomes, that's 23 from your dad. And so we would have to multiply that by 2, and you would end up with 46 chromosomes. That's 2N. All right, so let's go through this with not so many chromosomes. In G1, when a cell is first created, it has a setup like this, and it's going to go through its general functions, whatever its day job is, and the DNA is loose and unraveled. It doesn't really look like this. This would be condensed DNA. And it's going to go through G1, and then it's going to decide that it's time to divide. And in order to divide, we're going to need 2 copies of all the DNA, one copy to go into each of the cells. So we're going to have to duplicate our DNA. When we duplicate our DNA, each of these turns into this sort of X-shaped structure. And they are genetically identical. So you can see you've got the same number of beads, you've got the same arrangement of beads, and you've got the same alleles of the genes. It's important to remember that even though we've gone through S phase and we've replicated our DNA, we still only have 4 chromosomes. And the reason for this is that in our model, we're using magnets to represent our centromeres. But in real life, you would only have 1 centromere holding together the sister chromatids. And so when we count how many chromosomes are in a cell, we count them by the number of centromeres. And so in real life, this would be 1, 2, 3, 4 chromosomes. Even though we've essentially doubled the amount of DNA in the cell, they're still arranged as 4 chromosomes. 2 copies of chromosome 1 and 2 copies of chromosome 2. These 2 copies are called sister chromatids, and they are genetically identical. We created them during S phase using semi-conservative replication. After S phase, each chromatid has a sister chromatid, which is identical. Remember that the non-identical corresponding chromosome is called a homologous chromosome. After S phase, the cell will go through G2. At some point, it's going to be ready to go through mitosis. And this is the part where we're going to separate out all this DNA so that you can get the correct number of copies into each daughter cell. Mitosis, we're going to be making genetically identical daughter cells, 2 of them. So the first thing that happens in mitosis is prophase. I'm going to write the phases up here. I always think of them as PMAT, P-M-A-T, P being prophase. Some people like to say probably means another test as a way to remember it. But in prophase, a couple of things are going to happen. The first thing is that the nuclear envelope is going to start to break down. So instead of the nucleus being a protected area and the DNA being in there, there starts to be gaps in the nuclear envelope. The other thing that's going to be happening is that the DNA is going to condense. Now, we already show it condensed here, but in the rest of the cell cycle, G1 and G2, the DNA isn't all tight and coiled up because we're using it to make proteins. But during prophase, the DNA gets coiled up into these X-shaped structures. And the last thing that's going to be happening is that the centrioles start to separate. And the centrioles are little structures that are going to start making the mitotic spindle. So they're going to start sending out spindle fibers that are going to attach to our DNA. In metaphase, the nuclear envelope is completely gone, and our centrioles are on opposite sides of the cell, and they are sending out the spindle fibers. And what happens is a spindle fiber from one side pulls on one side of the centromere, and the spindle fiber from the other side of the cell is pulling on the other side of the cell. And so these guys get pulled back and forth until they line up here on the equator of the cell. And this is called the metaphase plate. I always think of metaphase as middle because they all line up in the middle of the cell. You can see there's no nucleus here. And as metaphase ends, we're going to get a separation of our sister chromatids, and that's in anaphase. Anaphase means these spindle fibers are shortening. Oops. And as they shorten, they're pulling our chromosomes away from one another. So the sister chromatids separate in anaphase. Telophase is our last phase. And telophase is essentially the opposite of prophase. So we've pulled all of our chromosomes to this side. We've pulled all of our chromosomes to that side in anaphase. And now what we're going to be doing is we're going to reform our nuclear envelope. So we're going to have a nuclear envelope that's starting to reform over here on this side of the cell. And we have a nuclear envelope that's starting to reform on this side of the cell. And our chromosomes are separated. And if you look carefully, you can see that you have 2 copies of chromosome 1 and 2 copies of chromosome 2 over here. 2 copies of chromosome 1, 2 copies of chromosome 2. And so we have genetically identical piles of DNA not only to each other, but to the initial cell that we started with. And as telophase progresses, the DNA will start to decondense. So remember, in prophase, the DNA condensed and the nuclear envelope broke down. In telophase, the nuclear envelope is coming back and the DNA is starting to loosen up again. The only thing that's left is something called cytokinesis. I think that's an I, kinesis. And this is the division of the cytoplasm. So everything else that's in the cell, all the organelles and things, are going to divide. So mitosis, PMAT, is division and separating out the genetic material. Cytokinesis is division of the rest of the stuff in the cell. And what you will see is the cell starts to kind of pinch in a little bit. So if this is the plasma membrane, it's pinching in. And eventually you're going to end up with 1, 2 little baby cells. Make sure you come back for the next video on meiosis. See you then.